Viruses hijack host cells, transforming them into factories that produce more of the pathogen. To survive, the immune system must identify and eliminate compromised cells. The body solves this problem using two specialized sentinels, CD8+ T cells and natural killer cells. The CD8 T cell operates as the precision sniper of the adaptive immune system. It requires explicit molecular proof of infection before it will engage a target. Conversely, the NK cell operates as the hair trigger patrol cop of the innate immune system. It relies entirely on instinct, attacking immediately when a cell's normal biological identification goes missing. This comparison table establishes their framework, the antigen-specific precision of the CD8 cell versus the non-specific NK patrol. Notice the exact trade-off with activation speed. The adaptive CD8 requires time, while the innate NK acts immediately. This raises a biological question. How do these two distinct targeting systems evaluate threats so differently? And how do they ultimately converge on the exact same molecular weapons to execute a target? Relying on a single recognition method would leave the human host completely vulnerable to any pathogen that mutates to hide its structural identity. Dual architecture ensures that evasion in one system automatically triggers the other. Target recognition occurs in a microscopic environment dictated by strict molecular rules, starting with the highly regulated adaptive system. This diagram illustrates the strict ID check logic of the CD8 T cell. The target cell must actively present a chopped-up viral or tumor peptide on an MHC class I molecule on its surface, which perfectly binds to the CD8 T cell receptor. Reaching this point requires several specific prerequisites. The CD8 cell needs prior priming from an antigen-presenting dendritic cell, a secondary CD28B7 co-stimulation signal, and expansion driven by IL-2. IL-2 is a signaling cytokine that drives the rapid division and proliferation of targeted white blood cells. The output of this rigorous, multi-step validation is a lethal, highly specific killer equipped with long-term memory for that exact pathogen. Shifting to the NK cell's domain, the biological priority is immediate suppression over specific memory. The NK cell completely discards the need for a specific viral antigen. Instead, it utilizes a missing self-detector to find abnormalities. Looking at the left side of this panel, we see the first specific trigger for an NK cell, the suspicious lack of an MHC class I molecule. Viruses frequently downregulate MHC expression to hide from CD8 cells, but doing so immediately alerts the NK patrol. The second trigger is the active presence of cellular stress ligands, notably MIC-A or MIC-B. MIC-A and MIC-B are specific stress-induced surface proteins expressed only when a cell undergoes severe infection or malignant transformation. The NK cell constantly weighs these inputs in a delicate balance. It actively measures inhibitory stop signals, like the presence of normal MHCI, against the activating go signals of stress ligands. The threshold for execution is entirely mechanical. The NK cell strikes automatically the moment the inhibitory brakes are removed. While CD8 cells wait for absolute proof of a specific crime, NK cells act immediately on suspicious absences. This creates a perfect biological trap for pathogens attempting to evade one system by triggering the other. Once a target is locked through either recognition pathway, both cell types transition to the execution phase using identical molecular arsenals. The process begins with the formation of the immunological synapse. The killer cell and the target cell membranes physically lock together, creating a tight, isolated junction between them. The killer cell releases molecules of perforin into this sealed chamber. These molecules insert themselves directly into the target's lipid membrane and self-assemble to forcefully punch open physical pores. With the door open, granzymes flood through these pores and enter the target cell. Granzymes are serine proteases, highly destructive enzymes that cleave peptide bonds to rapidly dismantle internal cellular proteins. Inside the target, the granzymes initiate a lethal cascade by forcefully activating caspase 3 and caspase 9. This cascade breaks the target cell structurally from the inside out. The mitochondria react by releasing cytochrome C. Cytochrome C is a vital mitochondrial protein that, when leaked into the cytosol, guarantees controlled cellular death, known as apoptosis. In this specific diagram, we see a second redundant kill mechanism shared by both sentinels, the Fas-L death handshake. When the killer cell's Fas ligand binds to the target's Fas receptor, it directly activates the Fad death domain. Fad stands for Fas-associated death domain, an internal adapter protein that bridges external signals directly to the Cas-base cascade, totally bypassing the need to punch holes with perforin. Beyond direct cell-to-cell execution, these sentinels deploy a third lethal vector, altering the surrounding biological battlefield. This graphic isolates the critical role of IFN-gamma, which is secreted heavily by both CD8 and NK cells. It acts as a massive amplifier, increasing macrophage lethality and forcing all surrounding tissue cells to upregulate their MHC presentation. Simultaneously, the release of TNF-alpha promotes intense localized inflammation, actively pushing any severely stressed neighboring cells closer to the threshold of apoptosis. By punching physical holes, triggering exterior death receptors, flooding the area with inflammatory signals, the host ensures the target cell has zero biological avenues for survival. Translating these microscopic mechanisms to macroscopic patient outcomes reveals exactly how these parallel systems dictate clinical life and death. According to this timeline of a successful immune response, NK cells rapidly handle early containment. They hold the line for the first few days, buying the necessary biological time for the CD8 cells to undergo clonal expansion and ultimately win the war. However, in cases of severe viral pneumonia, the devastating clinical hypoxia patients experience is often immune-mediated, rather than directly caused by the virus itself. This anatomical view details the damage triad occurring deep within the alveoli. CD8 cells aggressively destroy infected cells, NK cells eliminate the surrounding stress cells, and the release of ILH draws in highly destructive neutrophils. The immune system executes its cytotoxic protocols flawlessly. But in doing so, it physically obliterates the delicate structural integrity of the lungs, completely halting oxygen exchange. In oncology, tumor survival hinges on its ability to navigate and evade both sides of this dual architecture simultaneously. On the cancer immunity panel, we see the trap tumors face. Hiding MHC expression to evade CD8 snipers triggers an NK execution. Retaining antigens means CD8 cells destroy it. Progression only occurs when both biological systems fail. Finally, in cases of advanced sepsis, the breakdown occurs in a lethal cascade. It begins early with an unchecked cytokine storm driven by a massive excess of TNF-alpha. This progression chart maps the late-stage reality of severe sepsis. The early cytokine storm rapidly burns out the immune system, leading to clinical immunoparalysis. This state features complete CD8 exhaustion and severe NK cell dysfunction, leaving the patient defenseless against secondary infections. The integrity of human health relies entirely on the precise calibration of these two sentinels. NK cells kill what looks wrong. CD8 cells kill what is proven wrong.